Hello everyone, my name is Snehlata and I'm a PhD student and a PMI fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Respiration in Plants. So till now we have studied about the various phases that leads to the ultimately in achieving the goal of the respiration, that is energy production, right? So we have studied that respiration is of two types, aerobic and anaerobic. In anaerobic, we saw that there are mainly two types of respiration, while in aerobic, there is one, but it has certain steps. And those steps were what? Those were glycolysis, followed by link reaction, followed by uh, uh, Krebs cycle or TCA cycle, right? But we saw that this was the balance sheet till here, that in the glycolysis, we saw that the direct ATP production was only 2, while in the case of Krebs cycle as well, the direct ATP production was 2. So in total, till now, we have seen that there are 4 ATP production taking place. Apart from this, in this balance sheet, we saw that there are other sources of ATP production that is known as NADH and FADH2. And this NADH and FADH2 does not participate in ATP production directly. So what I mean is that these NADH and FADH2, they do not get converted into ATP, but they do something that leads them to get converted, that leads to the formation of ATP. Okay. And now we will see that out of this 38, since only 4 is related to the direct ATP production, 4 ATPs leads to the direct production. So how this 34 ATP is forming? Okay. So just for the revision, just remember that in glycolysis we form 2 NADH. In link reaction as well, we form 2 NADH. And in Krebs cycle, we form 6 NADH and 2 FADH2. Okay. Okay. So before moving to how ATP production actually takes place from NADH and FADH2, let's see that actually how many ATPs are formed by oxidation of one glucose. So in some book, you'll see that there are 38 ATPs. In some other cases, you'll see that there are 36 ATPs that are formed. So how is it possible? Okay. So what happens that this is your cell. This is your cell. Now in the cell, you have mitochondria. Your mitochondria have this inner invasion, right? So this is a mitochondria and it has inner. Okay. Now we know that glycolysis, where it takes place. The glycolysis, it takes place in the cytoplasm. Now what happens in glycolysis? Glucose leads to production of pyruvate, right? Pyruvate. Now this pyruvate, during this production, there are 2 ATP formation take place. 2 ATP and 2 NADH formation takes place. So it leads to the formation of 2 ATP and 2 NADH. But the process by which the formation of ATP from NADH takes place, it does not happen in cytoplasm, right? It happens in the mitochondria, and that too in this region that involve the participation of this periplasmic space and this inner mitochondrial membrane. Here we have F0, F1 particles. So which means that this NADH that is formed, it should get transported into the mitochondria. Now, the second problem is that in mitochondria, what we can see that it has membranes, right? It has 2 membranes. So how would this NADH get passed through these membranes? Okay. So we have seen different modes of transportation. In some cases, just the passive diffusion is sufficient, but in other cases, this certain molecules requires the carrier or the transporter to move them across the membrane. And this is also true in the case of this NADH. So we want this NADH movement from cytoplasm to mitochondria. So NADH should move should move from cytoplasm to mitochondria. Okay. Now, in order to do so, what happens? In order to do so, there are two shuttles. Like I said, that it requires carriers, right? So, in order to do so, there are two shuttles. The first shuttle is known as malate aspartate shuttle. Okay. So, to do so, there are two type of shuttle. The first one is known as malate aspartate shuttle. Okay. And the second type of shuttle is known as, and the second shuttle is known as glycerol 3 phosphate shuttle. It is known as glycerol 3 phosphate shuttle. So shuttle are what? They involve in transportation. Okay, these are the proteins that forms. You can see that moves from inside to outside, outside to inside freely. And if something gets attached to it, it will also leads to the transportation of that particular molecule. Just think it as a buses, right? So this buses, you go to a station, you sit in the bus, and then you move from one place to another in that bus. So similarly, these are the shuttles that act in a similar manner. So the compound will come and bind to it, and then they can easily transport across the membrane. Okay. Now, this different type of shuttle, these are basically responsible for how many total ATPs will be formed if a glucose is oxidized. Okay. So this malate aspartate shuttle, it is a good shuttle. Okay. Just remember it like this way. It is a good shuttle. So what it does, it takes the NADH, and then ultimately it transfer it as it is in the membrane. Okay. In this mitochondrial matrix. Okay. So it takes NADH and transform to matrix in same form. In same form. Okay. So and it takes NADH and it transfer it to the matrix in the same form. Okay. So what will happen then? Then 2 NADH will finally get if your shuttle is this one, malate aspartate. So I'm writing it as MA, malate aspartate shuttle. Okay. So what happens? That here it is NADH. Here also when it will reach, it will be NADH. But in the second case, what happens? This is a glycerol 3 phosphate shuttle. Okay. So this glycerol 3 phosphate shuttle, if this shuttle gets involved, then instead of NADH, it binds to NADH. Okay. But instead of NADH, it delivers FADH2 in the mitochondrial matrix. Okay. So it binds to NADH, right? In cytoplasm, binds to NADH, but deliver FADH2. Deliver FADH2. And how it does so, that's not important for now. So just remember, so if this glycerol 3 phosphate shuttle, it comes, so if G3P, glycerol 3 phosphate shuttle, it gets involved, then what happens? Then instead of NADH, here, what will reach in this cytoplasm mitochondrial matrix? It will be FADH. Okay. Now, if you remember that we know that in the case of NADH, from one NADH, how many ATPs can be formed? We will prove this in today's lecture, but in last class also, I told that from one NADH, 3 ATPs are formed. 3 ATPs can be formed. And from one FADH2, how many ATPs can be formed? It's 2 ATPs. So now we understood that why in some cases 36 ATPs are formed and in other cases 38 ATPs are formed. This is because this NADH, if it gets transported into matrix via malate aspartate shuttle, then NADH will lead to the ATP production. While if it gets transported through this G3P, that is glycerol 3 phosphate shuttle, then instead of NADH, it is a FADH2 that reach into the mitochondrial matrix, and that finally leads to the production of 2 ATPs. So it brings the gap difference of 2 ATP, right? For from one NADH it will be the one, and from two NADH it will leave to the gap of 2 ATP, and that's why you get 36 ATPs. Now, what can be the cases, or where in what cells actually this happens, that instead of NADH, FADH2 gets involved in the transportation of NADH. So it happens in the muscle cells and neurons. Okay, so this thing happens in the muscle cells and neurons where glycerol 3 phosphate is involved with the shuttle. Okay, so this brings us to the end of this Krebs cycle, and finally the ATP production. And now we can say that, or we understood that how 36 or 38 ATPs are formed. So in simple terms, you can write that why it is formed, because it depends on the type of shuttle used to transfer or transport NADH from cytoplasm to mitochondrial matrix. From cytoplasm to mitochondrial matrix. Mitochondrial matrix. Okay. So this is a situation where this number of ATPs depends on the type of shuttle that is used to transport NADH from cytoplasm to mitochondrial matrix. Okay. Okay. So with this, we have completed the Krebs cycle, and I have seen that how many ATPs are formed. Now we will see that how these ATPs are formed. Okay. But before moving to that, I just note one more point. So we know that in the case of Krebs cycle, if you go back and you'll see that there are various enzymes involved, out of which all the enzymes, okay, so I'll just write it here. So all the enzymes that are involved in the Krebs cycle are what? They are, just a second. So these enzymes are present in the mitochondrial matrix itself. Okay. So all the enzymes that are involved in the Krebs cycle, they are present in the mitochondrial matrix, except there is one enzyme known as succinate dehydrogenase. Okay. The enzyme is known as succinate dehydrogenase. And this enzyme, it is not present in the matrix, but this enzyme is present, or it is attached to the inner mitochondrial membrane. So suppose this is your inner mitochondrial membrane, right? So this enzyme is attached here. Okay. So this is your what? Succinate dehydrogenase. So succinate dehydrogenase, it is not present in mitochondrial matrix, but it is attached to inner mitochondrial membrane. Inner mitochondrial membrane. And this thing is important. Okay. So this is a different enzyme in terms of its location as compared to all other enzymes that are present in the Krebs cycle. So all enzymes, apart from this, all enzymes, they will be here in the matrix only. Okay, they will be in the matrix. But this enzyme, it is in the inner mitochondrial membrane. Okay. Okay. Now let's move to the electron transport system. So this electron transport system, basically it tells that how ATP production is taking place, or how the movement of electrons are involved in ATP production. Okay. So the electron transport system, as the name suggests, here what happens? Electron moves from one carrier to another. Okay. One carrier to another. And this movement, as we have also seen in the photosynthesis, that this movement ultimately leads to the formation of ATP. Okay, how? We will see shortly. Okay. So what happens due to this electron transfer movement? So it is responsible for the what? ATP synthesis. ATP synthesis. Okay. So since electron move from one carrier to another, it somehow leads to the formation of the ATP synthesis. Now, what is the location of this electron transport system? So the location of this electron transport system. So let's get back to the previous figure only. So this is your mitochondria, right? So the electron transport system, they will be located here. They will be located here in the inner not here. This is the crystal, but this part. Okay. So electron will located something like here. Okay. So what is the location? So location is inner mitochondrial membrane. Okay. So electron transport system they are located in mitochondrial inner mitochondrial membrane. Okay. Now this electron transport system it consists of, if we just talk about electron transport system, okay, so it consists of five complexes. Five complexes. Okay, five complexes. And what are these five complexes? So the first complex, its name is also first. We are presented by this one. So it is what? It is NADH dehydrogenase. NADH dehydrogenase. Okay. And this complex is what? This one is the largest complex. It is the largest complex. Okay. Now, the second is what? The second one is succinate dehydrogenase. So, have we heard this name before? Yes, right. So, succinate dehydrogenase is it is actually involved in Krebs cycle as well, right? So, it is one of the intermediate step of it. Actually catalyzes one of the intermediate step of Krebs cycle as well. But this it is also involved in the where in the electron transport system, and probably this is the reason why this is present or it is attached to the inner mitochondrial membrane. Okay. Now the third complex is it's known as cytochrome BC1 complex. Cytochrome BC1 complex. Okay. Now what is the fourth one? The fourth complex is cytochrome C oxidase. Cytochrome C oxidase. And the fifth one is F0F1 particle. So we have also heard about this F0F1 particle previously, right? So this F0F1 particle, it actually leads to the ATP production. So it leads to the ATP production. And this F0F1 particle is same as the F0F1 particle we have studied or you will study in the photosynthesis. Okay, there it was located in the chloroplast. Now here it is located in the mitochondria. Okay. Now let's see that now we have understood that what are the complexes that are involved in the electron transport system. So what happened? Basically, in simple terms, electron will move. The electron from here it will move to next, then it will move to next, it will move to next, and ultimately it gets accepted. Okay. So till here, like first to fourth. Okay. First to fourth. Here, what happened? Actually, the movement of electron takes place. But this F0F1 particle, it is located there, but here no electron transport takes place. Instead, here the hydrogen ion transport takes place that leads to the ATP production. So, you can also write, or in book it is also mentioned that if you talk about just electron transport system, so instead of 5, you can write 4. Okay. So if we talk about the oxidative phosphorylation, that how many complexes are involved, we can say 5, from including from electron transport till ATP production. But if we just talk about the electron transport system, meaning that the electron is getting transferred, so here there will be only first to fourth. So 4 complexes will be involved in the electron transport, not this F0F1 particle. Okay. Okay. Now let's see that how, like I said, that from one NADH, 3 ATPs are formed, and from FADH to 2 ATPs are formed. let's see how is it true. Okay. So for that, if we talk about NADH first, we talk about NADH first. So let's draw the this complexes. So how they are located. So this is a membrane. Okay. Right. This is a membrane, and this is the inner side of this. Both of these, by the way, are part of what? Inner mitochondrial membrane, right? Because here we know that all of these membrane, this membrane is also and double layered, and this membrane is also double layered. Okay, so I'm just zooming this part. Okay, this just think it as I'm zooming just this part. Okay, so this is the outer side, and this is the inner side. Okay, so here what we'll have? We have matrix. This region is matrix. This is inner membrane. This is inner membrane. And then this region was so intermembrane space. So by intermembrane space, what I mean? So just think it as this way. So this is a mitochondria, suppose. This is a outer membrane. Okay. This is a outer membrane. Now, this outer membrane is also what? Double layered, right? So it will have two membranes here as well. Okay. It will have two membranes like this here as well. Okay. So it is two membrane, but this is what? This is a outer membrane. This is outer membrane of mitochondria. Similarly, your inner membrane also, it is double layer. So it have this outer side and inner side. But between these membranes, this is one membrane, this is second membrane. This space is known as what? This is known as intermembrane space or periplasmic space. Okay. So let me erase this because I've just shown written to make you understand. Okay. So now, so we have understood that what this space is, where the space I'm talking about. So this space is basically this much, this space. This inner membrane space is this one. Okay. So this is your inner membrane periplasmic space, or it is known as periplasmic space. Okay. Or intermembranous space. Okay. Now the complexes that I said, first, second, third, fourth, fifth, they are located something like this. So this is your first complex. Okay. Now this one is your second complex. So this is your first complex. This one is your second complex. Now you will have third complex. So this is your third complex. And then there's cytochrome C. It will be located something like this. Okay. Cytochrome C. And then you will have your fourth complex. I'm not drawing fifth complex for now. Okay. So this one is your fourth complex, and this here will be your fifth complex. Okay. So what actually happens? So now we are talking about the NADH, that how from NADH the ATP production takes place. So in this case, the first thing that happens is now in the last reaction we have transported all the NADH to the mitochondrial matrix. Okay, so this is a matrix. We have transported all the NADH here. So this NADH, we know this NADH, it exists in this form, right? Now this NADH, what happens? It undergo oxidation. It undergo oxidation to form NAD+. Now oxidation is what? It is loss of electron. So here, from here, how many electrons will be lost? So here 1 and 2. Okay. So the electron that will be lost is 2. So these 2 electrons. Now these 2 electrons will come to complex 1. And in complex 1, it has various components that act as electron transport or electron carrier. So first of all, it goes to FMN. So FMN is a complex where it goes. And from FMN, it goes to FES. FES. Okay. So this electron, it is getting transferred. 2 electrons from here to here, here to here. Now, from here, what happens? This electron goes to another carrier that is known as UV quinone. Okay. So this is UV. I'm writing it as UV. So UV stands for UV stands for ubiquinone. Ubiquinone. Okay. UBIQUINON. And this ubiquinone is what? It is mobile carrier. So it can move. Okay. So it is mobile carrier. Okay. Now, from this ubiquinone, what happens? From this ubiquinone, it goes to your third complex. And your third complex involves what? It has cytochrome B. It has cytochrome that I'm writing by CYTB, cytochrome B. Then from here it goes to FES, and from FES it goes to cytochrome C1. Cytochrome C1. Okay, so it goes like this. From here, then here to 2 electrons here, 2 electrons here. Okay, and then from cytochrome cytochrome C1, it again goes to where? Cytochrome C. Okay, it goes to cytochrome C, and from cytochrome C, it goes to complex 4 component that majorly comprises of copper. So CuA. Then from here it goes to cytochrome A. Okay, cytochrome A. Then A3, cytochrome A3. Again, copper that I'm looking at CuB. And from here, it ultimately again come back to the mitochondrial matrix. Okay, so here this 2 electron, and when come back to the mitochondrial matrix, here in the presence of oxygen. So now we see the role of oxygen, right? So why oxygen is important? Because here this electron ultimately gets accepted by the oxygen to form what? To form water. Okay, and this water is what? It is called as metabolic water. This is known as metabolic water. Okay, so what does metabolic water means? It means that this water is formed upon metabolism of something. Okay, so this is not the water that we drink. Basically, it is formed inside our body, and hence it is known as metabolic water. Okay, so this is how the electron movement takes place. So electron, like here from cytochrome C, it goes to here. Okay, so this is how the electron movement takes place. In the case if NADH, if NADH is source of electron. Okay, if NADH is the source of electron, so this is how electron moves. So what we can see from here, what we can say that first of all, what different thing that we are seeing here. So one of the different thing we can see is the location of this cytochrome C. So this cytochrome C, although it is located on this inner mitochondrial membrane, but if you see all of these, they are located in between the two membrane. While this cytochrome C, it is located where? It is mostly towards the outside. Okay. So first of all, the important thing is location of this. Yeah. So the first part of it is important is the location of cytochrome C. So what is the location of cytochrome C? Location of cytochrome C. It is what? It is where? It is located in the outer side of the inner membrane. So located outer side of the inner membrane. Okay. So this is the location of cytochrome C. Now, what happens is with the movement of electrons, something else happens. Okay. So with the movement of electrons, something else happens. So what we are seeing here, that here this NADH upon oxidation, it is forming NAD plus. But along with this, it is also forming 2H plus, right? Because this hydrogen ion is also getting detached along with the electron. If you remove electron, the hydrogen ion will also get detached. So what happens? This NADH, as the electron moves, okay, the hydrogen ion also start moving, and they move from this matrix. They move. The movement will be like this. The direction will be like this. Okay. So with the movement of 2 electrons, 2 hydrogen ions move from this matrix to this periplasmic or intermembranous space. Here also, what is happening? The movement of electron is taking place, right? So here also, the hydrogen ion will move. So it will lead to the movement of the hydrogen, two hydrogen plus two hydrogen ion. Okay. And here also the electron movement is taking place. So what happens? Two hydrogen ions move. So this is the direction of health. So it is from matrix to pyroplasmic space. So as the electron movement takes place, it's not only the electron that is moving, but along with that, the hydrogen ion movement also takes place. Okay. So what we can write is, if a two electron, or simply you can say that two electron movement across electron transport system, across electron transport system, electron transport system leads to the, it leads to the movement of two hydrogen ions. Okay, two hydrogen ions from where? From matrix to intermembranous space to intermembranous space. Okay, intermembranous space, and this will lead to the formation of ATP. That we will see how. But okay. So now we are seeing what, that in NADH, what in the movement of electrons from NADH, what complexes are involved. So if you look at this figure properly, you can see that this what you say complex 2, right? This complex 2, it is not getting involved in the electron transport system. So in the case of NADH electron movement, with what complexes are involved? So in case of NADH, complex 1, third, first, third, and fourth are involved in electron transport. And similarly, if the electron is getting transferred, so, and it will also lead to hydrogen ion transport and hydrogen ion transport or transfer. Okay. So in this case, what complexes are involved? Complex first, third, and fourth. Okay. And NADH finally leads to how many movement of hydrogen ions from matrix to peripheric space. So we can see it leads to the movement of two hydrogen ion here, two hydrogen ion here, two hydrogen ion here. So one NADH, it leads to the transfer of, six hydrogen ion from matrix to intermembranous space, that I'm representing by IIMS. Okay. So this is what happens in the case of NADH. Now the next question is, what happens in the case of FADS2? So if you have FADS2, in case of FADS2 electron movement. So what happens in this case? So similar thing. This is a membrane. You have different complexes that we know. Okay. So we have complex 1, we have complex 2, we have complex 3. Right. I'm not drawing the components in between, but we have here cytochrome C and complex 4. Right. Complex 4. So what happens in this case? In this case, instead of this complex 1, right? This is complex 1, this is complex 2. So instead of complex 1, in this case, this complex 2, it is the primary electron acceptor. Okay. So in the case of FADS2, the primary electron acceptor, or the first electron acceptor, is what? Complex 2, not complex 1. Okay. So here this FADS2, it gets converted into what? FAD plus, and it leads to the formation of two electrons. And these two electrons get transported into the what? To complex 2. Okay. And now here we have what? We have ubiquinone represented by WB. So this ubiquinone accept the electrons. It transfer the electrons to the this complex 3. Okay. And this complex 3, what it does, we know that we have various things here. So electron get transferred from there to there to there. And then ultimately it moves to where? It moves to cytochrome C. Okay. This is what? This is cytochrome C. And from cytochrome C, it moves again to complex 4. Okay. So it is moving, it moves. And ultimately, this electron how many electron movement is taking place? Two electrons, right? So this two electron ultimately it does, it in the presence of oxygen and hydrogen ions, it leads to the production of water. And this water is what? It is metabolic water. Okay. This is also metabolic water. So wherever water is formed by the process of metabolism, that water is known as metabolic water. Okay. So in this case, what happens? So although in this case the complex 2 is getting involved, but from complex 2, the transport of hydrogen ion cannot take place. Okay, because this complex 2, it gets end here, right? It is located in the between the two membranes of the inner membrane, right? So suppose the hydrogen starts moving from here, then it will reach here. Then where it will go? It will get stuck, right? So it will need a proper channel from which it can transport across the membrane. Okay. So here this does not get involved in the transfer of hydrogen ions. Okay. So here the hydrogen ion takes place where? So it takes place where only two complexes. And these complexes are what? These are, this is one complex where the two hydrogen ion takes place, movement takes place, and this is the another complex. Okay. This is the another complex. Okay. So what we are seeing here in this case, for electron transport. Okay. First of all, for electron transport, what complexes are involved? Complex second, complex third, and complex fourth. Okay. But for hydrogen ion transport, for hydrogen ion transport, what complexes are involved? Complex third, involves complex third and complex fourth. Now we can see then in case of FATS2, total of how many hydrogen ion will move. So total hydrogen ion movement in case of FADS2, it is what? It is 4, right? 4 hydrogen ions. So in case of FADS2, total hydrogen movement is 4 hydrogen ions. Okay. So in now just remember, in case of NADH, it's 6, and in case of FADS2, it is 4. Okay. Now, next what we'll discuss, we'll talk about that how this hydrogen and electron ion movement ultimately leads to the formation of ATP. Okay. But before that, just one important thing to note. So the terminal electron acceptor, in the case of electron transport system in the mitochondria, terminal electron acceptor, what is it? The terminal electron acceptor is nothing but oxygen. Okay. It is oxygen. Okay. Okay. Now let's see how the ATP synthesis is taking place. So till now, we have understood that in the case of mitochondria, this is your inner membrane space. This is your inner membrane space. Okay. So till now, by the virtue of this electron transport, now the hydrogen ions have been deposited here in this periplasmic space. Okay. It are deposited in the intermembranous space or intermembranous space, or you can say periplasmic space. Hydrogen ions are deposited. Now, next thing is what? This next thing is again the pumping of this hydrogen ion back to the mitochondrial matrix. And when this pumping takes place, it leads to the formation of ATP. It leads to the formation of ATP. Now, let's see how. Okay. So, this is just for idea that where these things are taking place. Now, before erasing this, now we are seeing this crystal, right? This crystal here, similar thing will happen here. So this crystal has this particles attached to it. This is known as what? F0, F1 particles. These are known as F0, F1 particles. Okay. So now let's see what is happening here. So let's erase this. Okay. Let's erase this and just draw the F0, F1 particle. And that particle is also known as what? Complex 5. So now this complex 5 is also located in the inner membrane. Okay. It is located in the inner membrane. So this is here complex 5 till now. Here 1, 2, 3, 4, and then comes 5. So 5 will be like, it will look like this. Okay. It will look like this. Now, what does this fifth complex do? So it has two components. The one is known as F naught, and the second one is known as F1. And because of this, it is known as F naught F1 particle. Okay. Now this F naught particle, it acts as a channel. So it acts as channel. Now, what is channel? The channel is simply a carrier, right? That do not require any energy for the transportation of compounds across it. So this NADH, this F naught, it acts as a channel. So basically, it transport the hydrogen ion from where? So this reason is what it is your intermembranous space. Intermembranous space. Okay, and this reason here is what? This is your mitochondrial matrix. Right? This is your mitochondrial matrix. So now we have accumulation of hydrogen ions here. Right? Hydrogen ion is accumulated here by the virtue of why? Because the electron transportation was taking place from where? From across the electron transport system from NADH and FADH2, and that leads to the pumping of hydrogen ion to this space. So now what happens? That this F naught is an act as a channel, and it is what? It is a integral protein. F naught is a integral protein. Okay, meaning that it is located. You cannot isolate F naught without disrupting this membrane. Okay, but it is also intrinsic protein because it is located inside. Okay. Now this F1. This F1, it is actually an enzyme. It acts as an enzyme, and this enzyme is known as ATP synthase. This is now we understand. Now we can see that ATP synthase is here. It means the ATP synthesis will take place here only. Okay, and this F1 particle is what? It is extrinsic. So I have drawn it little incorrect. It should be like extrinsic. So it is mostly located outside. Okay, some part will be between the membrane, and then it is mostly located outside. Okay, now what happens? This hydrogen ion. So through this entire complex, two hydrogen ion movement takes place at once. So two hydrogen ion move at once, and when it moves, it leads to the formation of this movement leads to the formation of ATP. It leads to the formation of ATP. Okay. So what is happening here? As we can see that with the movement of two hydrogen ion, one ATP production is taking place. So we can write with the movement of two hydrogen ions, one ATP production takes place. One ATP production takes place. Okay, one ATP production takes place. So now we understood this thing. This point is important. Now let's go back to our NADH and FADH2. So this is how ATP production is taking place. Simply, once this hydrogen ion that gets accumulated in the intermembrane space, they start coming back to this mitochondrial matrix. Okay, and when they come back, they cause the conformational changes in this ATP synthase or F1 particle that leads to the formation of ATP. So with two hydrogen ion movement, one ATP production is taking place. Now let's get back to our FADH and NADH, FADH2 and NADH. We were saying that one NADH leads to 3 ATP formation and one FADH2 leads to 2 ATP formation. So now if you see that in the case of hydrogen, where is it? So in the case of NADH, we had 6 hydrogen ion formation or 6 hydrogen ion movement, right? Now this 6 hydrogen ion, as they will move from here to here, they will lead to production of 3 ATPs, right? Because with two hydrogen ion movement, 1 ATP production is taking place. So Here we can write that NADH leads to 6 hydrogen ion pumping, 6 hydrogen ion pumping from mitochondrial matrix to inner membrane space, and that will ultimately come back. And when it will come back, it will lead to formation of how many ATPs? 3 ATPs, right? And for FADH2, what we can write? For FADH2, it leads to 4H plus ion pumping, and that will ultimately lead to 2 ATP production. 2 ATP production. So this is what I was saying, and this is how it gets proved. Okay. So what is happening here? Here, we studied about electron transport and ATP synthesis. And this ATP synthesis is what it is known as oxidative phosphorylation. It is known as oxidative phosphorylation because here what we are seeing that phosphorylation is happening, phosphorylation of ADP is happening, and oxidative because here oxidation reduction rates take place, right? The electron is getting transferred, and if it is getting transferred, it will be the oxidation of that particular substrate or molecule. Okay. So because of this, it is known as oxidative phosphorylation. Okay. Now, from this, what points can we conclude? So if we see here that, so we can write this thing that the number of ATP molecules that are formed depends on the electron donor, right? So if the electron donor is NADH, the number of ATP formation is 3. But if electron donor is FADH2, how many ATPs will form? 2, right? So we can write that number of ATP depends on nature of electron donors. Nature of electron donors. Okay, and here, so we had studied about photosynthesis and this oxidative phosphorylation. So what is the difference between these two? Okay, so in the case of photosynthesis, what we saw that their light was the main source by which the electron generation was taking place, right? In that case, light was the main energy by which electron transportation was taking place. While in this case, it's not light, it's general or simple oxidation reduction. Okay, so this electron movement in this case is due to oxidation reduction. It is due to oxidation reduction. While in the case of photosynthesis, what it was? The case of photosynthesis, in photosynthesis, this movement was due to light. Movement was due to light. Okay. So here, no light is involved. Here, it is simply oxidation reduction. While in photosynthesis, it was due to light. Okay. So with this, we have completed this chapter, that is respiration in plants. So before moving, before ending this chapter, let's see, there are a few things that is important. So we have studied about the RQ value, right? Respiratory quotient in the first lecture itself. So in that, I told the respiratory quotient of few, like majorly of the of carbohydrate, fats, and protein. That was what? For carbohydrate, it was 1. For protein, it was 0.9, and for fat, it was 0.7. But apart from this, we have another source of energy that is known as organic molecules. So in organic molecules, the respiratory quotient is, in case of organic molecules, the RQ is greater than 1. Okay. Now, apart from this, if we talk about the succulent plants, in the case of succulent plants, what do we have? In the case of succulent plants, in night time, okay, in night time, the succulent plants RQ, it is 0, while in daytime, the RQ is 1. So it is greater than, sorry, not 1, greater than 1. Okay, it is greater than 1. So this is about the respiratory quotient that I missed previously. So just remember these values. And apart from this, one important thing that we called the respiration as a amphibolic process. So it's not a only catabolic process or only anabolic process. So why did we say that? So why did I say that respiration is amphibolic? Respiration is amphibolic process. Amphibolic process. So why this is the case? Because this whatever intermediates we have seen that respiration is a very large process. Okay, so it has various intermediates in between, and these intermediates go somewhere and leads to the formation of building blocks. For example, we know that in the case of Krebs cycle, we had alpha ketoglutarate, right? We saw that alpha ketoglutarate that was formed where? It was formed in the Krebs cycle, right? This alpha ketoglutarate is a source of amino acids. It is source of amino acid formation. Okay. So alpha ketoglutarate gets converted into glutamate. Okay. That is an amino acid. So it is what? It is anabolic process. So till now what we have seen the breaking of things. But these intermediates that are formed that ultimately leads to formation of some building blocks or some biosynthetic compounds. And hence it is also anabolic process. And together it is known as amphibolic process. Okay. For example, this succinyl CoA, it is also part of Krebs cycle, right? Succinyl CoA. So this succinyl CoA, it is part of only to the formation of chlorophyll, cytochrome C. Okay, cytochrome C. So these are few examples. Okay, with this we have completed this chapter. Now before ending, let's see few questions. So the first question is, which enzyme converts sucrose into glucose and fructose? So we have studied like this thing here in the case of glycolysis, that glycolysis, the major product is, the major substrate is glucose or fructose. But if sucrose comes, it first gets converted into glucose and fructose. And which enzyme does so? So it gets converted by the enzyme known as invertase. So the correct answer is invertase. Now the second question is, what is the end product of anaerobic respiration? So anaerobic respiration, we have seen it does not involve the oxygen. It has 2 or 3 steps based on its type. But what are the products that are formed? So respiration hai, so it will lead to the formation of carbon dioxide. So see where the carbon dioxide is. Here is carbon dioxide, here is carbon dioxide, and here is carbon dioxide. Okay. Now, apart from this, what else it forms? It forms pyruvic acid. That is right, but it's not the end product, right? That pyruvic acid or pyruvate gets converted into something. So the pyruvic acid option is not correct. Here is pyruvic acid. First is pyruvic acid. The option is not correct. Now what we have? We have carbon dioxide, water, right? Carbon dioxide in water forms. But if you see it's a C option, it is alcohol, carbon dioxide, and water. And we have seen that in the case of alcoholic fermentation, the alcohol form ethanol, right? So the correct option is C. C. Okay. Now, question 3 is the number of oxygen molecules required for glycolysis of one glucose molecule. So just look at the question carefully. Here it is asking what a glycolysis. So in the glycolysis, how many oxygen molecules are required? So in glycolysis, have we used any oxygen molecule? The answer is no. It does not involve the glycolysis. It does not involve oxygen and carbon dioxide. It is important for respiration, oxygen and carbon dioxide, but it is not involved in the glycolysis process. So the correct answer is 0. Okay. Okay. So what is the next question? The process of conversion of glucose into alcohol is known as what it is known as. So this process is known as respiration. No. Digestion. No. Fermentation. Okay. So this process is known as fermentation. Okay. When glucose gets converted into alcohol, it is a type of respiration, but the process is known as fermentation. And this is what? This is anaerobic process. This is what? This is anaerobic. Okay. Now, the next question is respiration is a endothermic reaction, photodecomposition reaction, electrochemical reaction, exothermic reaction. So respiration leads to the release of energy, right? So it's exothermic. Now, proton accumulates on the which side of the mitochondria? So proton means what? Hydrogen ions, right? Proton is what? Hydrogen ions. So where they get accumulated? So we have seen just now we have seen. So it gets accumulated. It gets accumulated. Where is that diagram? Yeah, it gets accumulated here, right? Here. So in this space is intermembranous space. Okay. So this space is what? Intermembranous space. So it gets accumulated in the intermembranous space. Okay. Now, the products of Krebs cycle essential for oxidative phosphorylation is okay. So, what products are important for oxidative phosphorylation? So, oxidative phosphorylation is ATP production. So, what are the components that we just saw? Is it NADPH and ATP? No, ATP is not required for oxidative phosphorylation. Is it acetyl CoA? Acetyl CoA is important in the first of all, it is important in the Krebs cycle, but not in the oxidative phosphorylation. So this is incorrect. The next is CO2 and oxaloacetate. No, we did not involve these two. We just saw. The next option is NADH and FADH2. And this is the correct answer, right? Because NADH and FADH2 leads to the hydrogen ion pumping or proton pumping and then formation of ATP. Okay. The next question is DAS accepts hydrogen from malate. Okay. So malate is where we have studied about malate in where in the case of what you say a Krebs cycle, right? In the case of Krebs cycle, we saw that malate is involved, but from malate, what was the compound that accepted the electron from malate? If you remember, what was that? So from malate, the electron that got accepted is just a second. It is FAD or NAD. We do not have NADP here involved. We do not have FMN involved here, right? So from here, malate, the electron is getting accept. What happens to malate? So malate we have from malate we form oxaloacetic acid, and here the compound that is involved is NADH, NAD, right? And it accepts electron to form NADH. So here the answer will be NAD. Okay. Okay. Now the last question is the acetyl CoA is formed from pyruvate by what reaction? Okay. So acetyl CoA, we had pyruvate. Then we have acetyl CoA. So we saw that the series of reactions takes place here. What are those reactions? Here it is 3 carbon and it leads to the formation of 2 carbon molecules. So it was first of all decarboxylation. Second, this pyruvate was undergoing oxidation to form acetyl. It was oxidation. So what will be the correct answer? Dehydration, no. There is no release of water. Reduction. Reduction was of NADH, right? NAD to NADH. So it was not reduction of pyruvate. So this is also incorrect. The third option is oxidative decarboxylation. Yes, it takes place. The pyruvate undergo oxidation and it undergo decarboxylation. So the correct answer is oxidative decarboxylation. And what about the fourth option, dephosphorylation? No, because here no phosphate group is involved. So the correct answer is oxidative decarboxylation. So with this, we have completed this chapter, that is respiration in plants. That's all. Thank you.